We talked about cost as a factor. Decreasing costs, for example, in genome sequencing comes to mind. Development of automated cell processing systems to generate highly pure, reproducible, and consistent population of cells. Improvements in computational technology is another item that needs improvement.
Whereas we have developed these drugs very specifically in the preclinical phase, we have very few tools to allow us to actually screen which patient could benefit from this precision medicine. So this precision medicine is not very precise at all. And there I think we are going to see a big difference, because now we have models that are specific to patient, and we have the drug developers to chemistry to make the compounds that are required for it. So I think indeed now we will really be starting to work on precision medicine.
There's a lot of unmet needs when we're thinking about kind of where the organoid research is going and how it's being utilized. A few of them, include really developing new technologies to allow us to kind of rapidly screen organoid sensitization and resistant mechanisms. Others include looking at really kind of how's the heterogeneity of the organoid culture is changing and how is it evolving and adapting.
We hear a lot about using organoids and 3D models for drug screens, and that's what we're interested in doing also. But as they say, the proof is in the pudding. So being able to show that your preclinical screens and organoids actually translate into true efficacy in the clinic would be amazing. And I think that's where we really need to go in terms of translational medicine and improving precision medicine.
To get prescribed a drug, you need to show that it'll have a benefit to the patient. And so a lot of times you'll wait until a very late stage in the patient's course of treatment to give them a drug as like a last ditch hope. But instead, if you have patient organoids, that could be your biomarker. So you can prove from the get-go, this patient will respond, should respond, and then get approval to get the drug.
While it's great that we can develop these huge banks and run all these screens, the key to going from using that to inform a targeted clinical trial to actually informing patient care is something that is sort of waiting. And the only times that we use it now are in areas of compassionate use, or when there's an FDA approved drug that an insurance company is willing to pay for, because a tumor has a specific mutation that it shouldn't. BRCA mutation in a pancreatic cancer case. The ability to scale and sort of correlate our actual responses is really the key missing agent.